I'd like to thank Corteva and Texas A&M for the opportunity to be here today, and I'm happy to be here as the representative for Virginia Tech's Soybean Breeding Program and our work that we've been doing using CRISPR to develop low trypsin inhibitor soybean lines. So soybean is highly valued for its protein content and amino acid profile and nutritional benefits. And because of this, as you can see here, about 76% of global soy production goes directly towards animal feed. But if you look at the bottom of that percentage, you'll see that only 7% is consumed raw or goes directly to livestock. The majority has to be first processed into soy cakes or meal. And this is done using a very costly and time-demanding heat treatments, and this is to denature trypsin inhibitor. So trypsin inhibitor, which I'll be referring to as TI through the rest of the presentation, is an anti-nutritional factor that occurs naturally in soybean, and it causes severe gastrointestinal issues and can reduce growth rates up to 30%. However, it's present in all plant tissue and is natural for the plant's natural pest defense. And because of this, it's difficult to focus on reducing trypsin inhibitor in the seed alone while leaving the rest of the plant tissue untouched using traditional breeding methods. So the work that a previous student did in 2021 was to use CRISPR-Cas9 to delete 2 genes specifically responsible for the expression of trypsin inhibitor in the seed. And so here you can see the binary vector that was used for the 2 genes, which were KTI1 and KTI3. So the alignment of the KTI1 mutants, there was a 66 base pair deletion for the first gene. And for KTI3, in several of the lines, we saw a 69 base pair insertion with a 22 base pair lost. We also saw some lines where there was only a C deleted, and other lines where there was a 7 base pair lost. And so with KTI3, we saw frame shift mutations. Now, Williams 82 was used as our wild type genotype for this study. And as you can see here, TI quantification in the seed for Williams 82 is just under 5 milligrams per gram in the seed. But with the 2 gene deletions, that reduction was to less than 1 milligram per gram in seed. And so it was one of those lines with the 2 gene deletions that we decided to advance, and which we have named VT5-26. And so the objective of this specific study was to evaluate the agronomic performance of this line compared to the wild-type Williams 82 used in its development. So this was done in a 2-year field study in 2 locations in Virginia. Blacksburg, which is up in the Appalachian Mountains, and the Blue Ridge is our main campus and main research station. And then one of our AREX, Warsaw, is out there on the coast, and so 2 very different environments. And we were looking specifically, again, at VT5-26, Williams 82, and then we used AG3803 as our commercial check in the study. And the pictures there on the right, the top one is about 1 month of growth. The VT line is the second one, if you're looking towards the back of the image, second plot. The middle is approximately 2 months of growth in the field, and then the last was right before harvest. So because of TI's importance towards pest defense, we also performed a pest evaluation of the study. And so we were looking both at leaf feeding damage as well as pod feeding damage. And so for leaf feeding damage, we used a standardized scale. We took 5 leaves at random from the plot and visually investigated or decided the damage, as you can see there on that image. And for pod feeding damage, we took 100 pods from the outer 2 rows so as not to affect the final yield count. And we were looking for signs of any feeding, which hard to see on that image, but they're little black marks on the outside of the pod, which is usually from stink bugs. That's what we saw most in the field. And we were looking to see if they corresponded with a shriveled or misshapen seed on the inside. We also investigated response to soybean cyst nematodes, and this was done by inoculating plants in a controlled greenhouse environment. And so we took an average of 12 replications from that to calculate nematode cyst count per plant. And so for our results, for your typical agronomic traits, we saw no significant difference in performance between VT 5-26 and Williams 82. As you can see here, maturity, height, and lodging were essentially the same. There was a small reduction in yield, but it was significantly statistically not significant in this study. When it came to the pest feeding damage, there was a slight increase in susceptibility in the VT 5-26 line, but again, it was determined to not be statistically significant for the study. In terms of seed composition, there was no difference in oil percentage. There was a 1% decrease in protein. However, this was expected because trypsin inhibitor is a protein, and so we've reduced it in the seed. And this 1% loss is negligible compared to the 5 to 20% protein, which can be lost during those heat treatments that I mentioned earlier to denature TI. However, our most interesting result for this study was the nematode cyst count. We saw a significant reduction in the number of cysts per plant on the VT 5-26 line, more than 100 less cysts per plant compared to the wild type. And there have been a few studies that link trypsin inhibitor to nematode resistance. And so this is something we're interested in looking into in future studies to see if there is a correlation between the genes we deleted and nematode resistance. And so our conclusions from this study was that we did successfully knock out the 2 genes responsible for TI in the seed, and that there was no statistically significant difference in agronomic performance between the 2 lines. And so the results validate that CRISPR-Cas9 is a promising tool for reducing TI in soybean and paves the way for the development of crops better suited to meet global food and production challenges. And our next goals will be to incorporate these 2 gene deletions and higher performing lines to better benefit producers. And with that, I'd like to thank the Virginia Tech soybean team for their continued support, as well as the Virginia Soybean Board for the funding of this project. And with that, I'm happy to take any questions.
Thank you for your talk. So you showed a reduction in trypsin in the knocked out lines, but it's not a complete, you didn't completely get rid of it. So what is the amount of trypsin in soybean meal after it's been processed? And is this low enough to not have to go through that process anymore?
Yes, so those heat treatments I mentioned earlier typically reduce it to about 2 milligrams per gram. And so the remainder that you saw in that graph, so there's Kunitz trypsin inhibitor, which is what we deleted here in this study, and then there's Bowman-Burke trypsin inhibitor, which is a much smaller percentage of the seed, and is something we could look into deleting as well in the future. But that's what you saw in that graph. It was the Bowman-Burke that remained. Thank you for your question.
Thank you for your talk. I really liked it. I had a question about the insect damage and how you showed that there wasn't too much going on in this experiment. I was curious what your thoughts were. If this mutant were to be deployed in the field, do you think there'd be a risk of when you grow it en masse, it would attract insect, it would have any change in that immune system would result in a greater problem?
So I don't think there would be an increased effect in large production because this was grown on multiple acres with other research studies. And we think that if you deployed this in production settings with an insecticide, that it would kind of eliminate any risk of that. Thank you.
Let's thank our speaker.
Thank you.